and in collaboration between Agroscope and Bologna. And today she will speak about the impact of intrauterine growth restriction on pig growth and microbiota. So you can start.
Hello, everyone. Can you hear me well? And can you see the screen?
Yes.
Okay. So, but it's okay. So we know that the economy of the modern pig production system relies largely on the reproductive performance of the sows. And that's why in the last decades, the genetic selection in this sector focused on increasing the number of piglets born per litter, so the prolificacy of the sow. And this actually worked because now we have hyper-prolific sow lines that can give birth up to 18, 20, even 22 piglets alive per litter. So we saw in the last years an increase in litter size, but also we saw an increase in the number of piglets that are born with a low birth weight and piglets that are exposed to intrauterine growth restriction or IUGR. IUGR is defined as the failure of the fetus to reach its genetically expected growth during gestation, and it results in the impaired development of its organs. And the main cause of this condition is insufficient space within the uterus, which is a direct consequence of the genetic selection for prolificacy. So the genetic selection increased the number of piglets, but actually there is not enough space in the uterus because the uterine capacity was not increased. And so we have a situation of intrauterine overcrowding that is associated with reduced placental weight per fetus and reduced uterine blood flow per fetus. So some of the piglets receive a reduced amount of oxygen and nutrients, and they develop poorly. And those are the IUGR piglets. Those piglets, to survive during gestation, they have to redistribute the blood flow to the brain. And this mechanism is called the brain sparing effect. So basically the development of the brain is prioritized compared to the other organs. And this results in the fact that at birth, those piglets have a brain which is relatively bigger and heavier compared to the rest of the body. And one of the organs that is most affected by the blood deprivation is the liver. And that's why the gold standard to diagnose this condition is an increased brain to liver weight ratio at birth. And we know from the literature that this condition compromises the development of the gastrointestinal tract. There are several studies which reported a reduced length and weight of the intestine, but also reduced villus age, increased crypt depth, and a different transcriptomic and proteomic profile in the intestine of IUGR compared to normal pigs. And also we know that this condition affects also the microbial colonization of the gastrointestinal tract. So also in this case, we have several studies which reported a distinct taxonomic profile in IUGR compared to normal pigs at different developmental stages. Also, I told you before that during the IUGR pregnancy, the fetus receives a reduced amount of oxygen and nutrients, including glucose. So it has to initiate some metabolic and endocrine responses to utilize other sources of energy, like the fatty acids. Indeed, one of the responses is an increased adipocyte proliferation. Increased adipogenesis means that we have an increased capacity to store fatty acids that then can undergo oxidation and provide energy for the fetus. And this is a mechanism that it increases the chances of survival of the fetus in a situation in which there is a poor or intermittent nutrition, like in the IUGR pregnancy. And some studies suggested that this increased adipogenesis lasts also in the postnatal life. And this seems to be confirmed by findings at the blood metabolome level. There are not so many studies in this field, but from the few studies that we have available on human neonates and piglets, what emerged was an early pattern of glucose intolerance, insulin resistance, and altered lipogenesis and lipid oxidation in IUGR compared to normal neonates or piglets. So considering the modification that IUGR imposes on the development of the gastrointestinal tract and on the microbial colonization of the gastrointestinal tract, and also the endocrine and metabolic adaptation that we have during the IUGR pregnancy, in our study, we hypothesized that we would have found clear differences both at the fecal microbiota and blood metabolome profile between IUGR and normal pigs at different developmental stages. And we believe that microbiota and metabolome analysis hold the potential to identify biomarker of this condition. If we identify biomarker, we can get new insights into the complex mechanism behind this condition, and we can start to develop an effective treatment. This would be really useful because this condition can affect up to 30% of piglets in a litter, and it's associated with increased mortality, morbidity, stunted growth, and reduced feed efficiency. So it's also a problem from the economic point of view. So in our study, we had 168 piglets, and on day one, we performed computed tomography scan imaging to assess the weight of the brain and the liver and the brain to liver weight ratio. Because as I told you before, the gold standard for the diagnosis, it's an increased brain to liver ratio at birth. Then we selected 24 piglets. The 11 piglets with the highest ratio were assigned to the IUGR category, and the 12 pigs with the lowest ratio were assigned to the normal category. Then we followed those pigs until the end of the production cycle. We recorded the growth performance in terms of average daily gain and feed intake. We assessed the body composition at different developmental stages using DEXA. We collected feces to perform microbiota analysis and blood to perform plasma metabolomics. Regarding the growth, we weighted the pigs once a week, and then we calculated the average daily gain for the lactation, starter, grower, and finisher periods separately. Also, we recorded the feed intake individually from 20 kilos of body weight until slaughter, and we calculated the gain to feed ratio from 20 kilo to slaughter. For the body composition, we performed DEXA at weaning, at the start of the grower period, and at the end of the finisher period to assess the body composition. The output of DEXA is the total bone mass, the bone mineral density, the total lean mass, and total fat mass. For the feces and blood, we sampled feces and blood during the lactation period around day 16. And this is our first time point, and it was right before the crib feed administration. Then we sampled again feces and blood at the end of the starter period, and this is time point 2. And we made the sampling right before the grower diet administration. And then we sampled just the feces at the beginning of the finisher period, right before the finisher diet administration, and this is time point 3, and at the end of the finisher period, right before slaughter, and this is time point 4. So if we look at the body weight, we performed the sampling approximately at 5 kilos, 20, 60, and 110 kilos of body weight. The feces were collected directly from the rectum and then were stored at -80 before the analysis. For the analysis, we extracted DNA from the feces, and then we amplified and sequenced the DNA at the V3, V4 hypervaluable regions of the 16S rRNA gene. Once we obtained the results, we analyzed the alpha diversity in the two groups, IUGR and normal, the beta diversity, and we performed an analysis of taxa. For the blood was collected from the jugular vein and then was stored in ice before centrifugation. Then it was centrifuged to obtain the plasma that was used to perform proton nuclear magnetic resonance analysis. With this analysis, we obtained the concentration of the metabolites in the blood, and then we compared the concentration of each metabolite between the two groups. Then we used PCA as unsupervised multivariate analysis for outlier detection, and OPLSDA as supervised multivariate analysis to check the spontaneous grouping of the samples according to the category and to identify the metabolites of interest that were defined as those with a VIP above 1 and absolute high loadings. Those are the results of the grow performance. So here we have the average daily gain in the lactation period. We can see that there is the brain to liver weight ratio on the x-axis and the average daily gain on the y-axis. And on the left, we have the normal pigs with a low ratio. And on the right, the IUGR pigs with the high ratio. And it's clear that the IUGR pigs have a lower average daily gain, and the effect is stronger on the castrates. We have a similar situation in the starter period. Again, you can see that as the brain to liver weight ratio increases, the average daily gain decreases. And again, we have IUGR pigs that grow slower. A similar situation in the grower period. So again, IUGR pigs grow slower compared to normal one. But in the finisher period, there is no difference anymore between the two groups in terms of average daily gain. Here we have the gain to feed ratio. And again, we can see that as the brain to liver weight ratio increases, the gain to feed ratio decreases. So IUGR pigs were less efficient in utilizing feed compared to the normal ones. And here we have the age at slaughter. This also it's quite important. You can see that IUGR pigs were quite older at slaughter. In this study, they were actually 18 days older compared to the normal pigs at slaughter. This is really important from an economic point of view. Regarding the body composition, there was no difference in muscle and fat percentage at any of the time points. The time points, I remember it was were winning beginning of the grower period and end of the finisher period. But we found difference in the bone mineral density at weaning. You can see that the IUGR pigs had a higher bone mineral density at weaning, while the situation was the opposite at 20 kilos of body weight, where we have the IUGR pigs that have a lower bone mass and also a lower bone mineral density compared to the normal group. Those are the results of the faecal microbiota for the alpha diversity. So we can see that according to the Chow index, there was a significant effect of time on the microbiota alpha diversity, but there was no effect of the category except for a tendency at 9.2, that is the end of the starter period, in which the IUGR pigs had a tendency for a higher alpha diversity compared to the normal group. And we have the same situation when we use the Shannon index. Again, we have a significant effect of time and just a tendency for the category. While with the inverse Simpson index, there was no difference based on time or category. For the beta diversity, in the lactation period, there was a significant effect of the category and the litter of origin on the beta diversity. At 10.2, we had just a tendency for the category. And at 10.3, we have again a significant effect of the category and just a tendency for the litter of origin, while there is no effect in the finisher period at 10.4. This is the differential analysis of taxa, and we can see that at T1, during the lactation period, normal pigs had a higher abundance of Ruminococcus, while the IUGR pigs had higher abundances of Clostridium sensu-stricto and Rombustia. At 10.2, we have the normal group with a higher abundance of this bacteria that belongs to the Lactobacilliaceae family, while the IUGR pigs had higher abundance of bacteria belonging to the Brevodellaceae, Rickettsiaceae, and Lactospiraceae families. At 10.3, we have the normal group with higher abundance of Prevotella, while in the IUGR group, we have higher abundance of this bacteria that I'm not gonna pronounce because it's too hard, but it belongs to the Bacteroidetes order. And at 10.4, we don't have differences anymore between the two groups. those are the results of the plasma metabolomics. So at T1, during the lactation period, none of the metabolites was actually different between IUGR and normal pigs. Here we can see the results of the PCA, and you can see that there is no spontaneous grouping of the samples according to the category. And those are the results of the OPLSDA analysis. In this case, we can see that there is a spontaneous grouping of the samples according to the IUGR and normal category. So there is a good group discrimination, but actually the model had weak predictive ability and high risk of overfitting. At T2, no, sorry, this is the plot of the loadings of the OPLSDA analysis, and in this plot we can see the metabolites that contributed the most in the separation between the two groups. And leucine is the most important metabolite, and it was more abundant in the blood of normal pigs. But anyway, none of the metabolites had a VIP above 1, so none of the metabolites could be considered significant in the metabolic signature in the lactation period. At time point 2, so at the end of the startup period, the only different metabolite was asparagine, that was lower in IUGR compared to normal pigs, and we can see it in the graph here. Regarding the PCA, we can see that in this case there is a tendency for a spontaneous grouping of the samples, and again we can see in the OPLSDA that like before we have a good group discrimination, but also in this case the model had a weak predictive ability and high risk of overfitting. Here we have the plot of the loadings, and in this case the metabolite which contributes the most in the separation is asparagine. But again, like at time point 1, none of the metabolites, not even asparagine, had a VIP above 1. So none of the metabolites was significant in the separation between the two groups. So to summarize a little bit, from this study, we saw that intrauterine growth restriction has a negative effect on the average daily gain of pigs from birth until the end of the grower period. It doesn't have an effect on the lean and fat percentage in the body, but it has a dynamic effect on the bone mass and bone mineral density. If you remember, before I told you that at weaning there was an increase in the bone mineral density in the UGR group, while the situation was the opposite at 20 kilos of body weight at the end of the startup period. Here we had a reduction of the total bone mass and also the total bone mineral density. And this is actually in line with the human literature, in which we know that humans affected by UGR have higher risk of osteoporosis. And this could be also a problem in pigs from the animal welfare point of view, because those pigs could be at higher risk of fractures in the grower fusion period. We also saw that IUGR has a long-lasting effect on the fecal microbiota composition from birth until the end of the grower period. IUGR pigs had lower abundances of beneficial bacteria. At T1, they had a lower abundance of Ruminococcus, and we know that this bacteria belongs to a family that is able to ferment dietary fiber and produce short-chain fatty acids, which are important for inflammation, barrier function, and gut health. While at T2, there was a reduced abundance of bacteria belonging to the Lactobacilliaceae family. And for this family, it was demonstrated in pigs a positive association with energy and nitrogen efficiency. While at T3, there was a reduction of the Proboscideaceae family. And this family is like Ruminococcus, this bacteria can ferment dietary fiber and produce short-chain fatty acids that are important for gut health. So there could be an association between the reduced abundances of these important and beneficial bacteria and the fact that those pigs grew less until the end of the grower period. Regarding the metabolome, there was no difference in the plasma metabolomics between IUGR and normal pigs in the lactation period, while at the end of the starter period, the only significantly different metabolite between the two groups was asparagine. Asparagine was lower in the blood of IUGR pigs. Asparagine is a non-essential amino acid, and it can be a precursor of glutamine and glutamate, which are important sources of ATP in the enterocytes, but it's not involved in lipogenesis and lipid oxidation. And also other metabolites that are involved in these pathways and also in energy and amino acid metabolism were not affected in our study. And this is also in line with the fact that we found no differences in the percentage of fat in the body at any of the growing stages. So to conclude, we found a distinct taxonomic profile regarding the fecal microbiota, and I think and I hope that future research will identify other taxa that are consistent across studies and that can be utilized in the future as a biomarker of this condition. If we find biomarkers, we can start to develop a target and effective treatments to mitigate the negative effects of this condition. Thank you.
Thanks a lot for your presentation. It was very complete and clear. Is there any questions from the attendees? Cécile, you can go maybe turn on your camera if it's possible for you. Cécile, I see the end. I think you're muted. Or maybe if it's easier for you, you can also write the questions in the chat. Are there other questions?
Yeah. So I told you that the gold standard to identify this disease as it's an increased brain weight to liver weight ratio, and this is normally it's difficult to assess. So usually the diagnosis is just based on the low birth weight of the piglets or the head shape, because they usually have this bumped head shape. But in our study, we actually used this ratio, and we calculated the weight of the organs using computed tomography scan imaging. So we scanned all the piglets at birth on day 1. We assessed the weight of the organs, and then we calculated the ratio. And we selected the 24 piglets based on this ratio. The IUGR group were the 12 piglets with the highest ratio, and the normal pigs were the 12 piglets with the lowest ratio. So we really took the two extremes of the population.
I also have a question. Because at the end you speak about potential treatments, and are there already any treatments, and what could be? Do you have any idea how it could be treated?
Yeah, but right now there is no effective treatment, and this is also a consequence of the fact that usually they are identified based on the birth weight or head shape, which is not really accurate. So this is also a consequence, like if you cannot really diagnose them, it's difficult to find an effective treatment, and right now there is none. But based on these results, for example, a treatment could be the administration of probiotics or prebiotics to shape the gut microbiota. because we saw that they have lower abundances of bacteria that are actually important for the gut health and for the growth of the pigs. So this could be a treatment, or just a more like intensive care when they are born, like administration of colostrum or essential amino acids. Those could be treatments, but as long as we cannot really diagnose them and we don't know what exactly is the problem, it's a bit difficult to find a target treatment.
Yeah. And are similar treatments also done in humans, or do you know how they cure it in humans?
Again, there is no real treatment. Like you can do some like symptomatic treatment. You can administrate essential amino acid or like important nutrient, but there is no right now no real effective treatment that can mitigate all the negative effects of the condition.
Yeah. Thank you.
You're welcome.
Are there some more questions?
Yes, I have a question, but I cannot be able to type it in the chat. It's like it is restricted. I am a PhD student from University of Debrecen in Hungary, and I wanted to ask a question. Is it okay? I can go on.
Yeah.
Yeah. So I had actually 3 questions. The first one was what was the threshold or the base numbers that you are able to isolate the piglets from the whole group of 268, the ones that were able to make it to become IUGR and the ones that were able to make it to become the normal. That is, what was your base numbers, the exact numbers between brain and liver ratio. Then if I move to the second question, can I go on all the 3 questions?
Yeah.
So if I can be able to ask the second question, that is it standard that the farm operations only allows, let's say, castrated piglets to enter into the experiment? Because I've seen on your results that you have indicated castrated and then females. So there are no uncastrated. Then the third question, is that perhaps what is your industrial recommendation, especially we cannot be able to prophesy or know or diagnose the litter that is coming with IGR piglets. But what is your industrial maybe recommendations in case some litters can be even noted earlier, which could have such like an instance? Because it's quite necessary to understand what could be the implications if a litter is more biased having IUGR piglets. Thank you.
So for the first question, there was not a threshold, but as I told you before, we took the 2 extremes of the ratio, and the values were around 1 for the IUGR category and 0.35, 0.4 for the normal category. But we didn't set a threshold. We just took the 2 extremes, so the 12 with the highest and the 12 with the lowest, because we wanted really to work and to see the difference between 2 extreme groups. For the second question, I'm not sure I understood. You asked about castrated pigs, if it's allowed to, if you can repeat, because I'm not sure I understood well.
Yes, I was asking that in your sample, you indicated sex and you indicated castrated and then others were female. Is it a normal farm practice that only castrated animals can be available for an experiment, rather than having, let's say, the intact males also getting into the experimental treatments? Was that possible?
So in our farm, all the males are castrated. So when we do experiments, we actually have available females and castrates. Like it's a routine practice. Pigs are castrated, the males.
Okay, thank you. And what about the last question?
About the last question, what could be done? It's a bit hard to answer. Like from an industry point of view, what could be done is that if you're able to identify correctly the IUGR piglet litter, you can, for example, start to select and for the sows which produce less IUGR piglets and just keep them, or as what you could do, like from what I believe after my research, it's I think it's quite clear that there are like big differences in the at the microbiota level. So I could try some strategies based on the microbiota, so administration of probiotics and prebiotics, but this of course need to be tested like more accurately. But I would take that strategy, like select the sows which produce less UGR piglets in a litter and try to supplement with probiotics and probiotics.
Okay, thank you. Maybe just to add on something else as a concern, because if you are selecting sows that are producing more of IUGR piglets, could that be a way of also selecting for the sows that are still producing the runs or the smalls? Because in normal circumstances, you'll find that most of the IUGR piglets are actually the runs in the litters, the small ones. And therefore, by extension, that could actually be that you are selecting for sows that are producing runs. Is that industrial applicable?
No, I actually said the opposite, that you should select for the sows that produce less IUGR or runs piglets.
Okay. Thank you. I really appreciate. Could you be able to share the presentation? I'm interested, by the way, for knowing more about this.
Yes, of course. Yes.
I don't know how can you be able to share it, because we have not been able to maybe share our emails or something like that.
I think I can send it to the people who organize the meeting, and then they can share them. I don't know.
We also record the meeting, so you can also access the presentation like that, and we will share.
Okay, thank you so much.
Thank you.
There is a question in the chat.
Yeah, so it is related to large litter. It's actually the main consequences of large litter, and that's why at one point we should question about the fact that is it actually worthy to keep increasing the litter size, is if we also increase at the same time the number of those affected piglets, because they have higher mortality rates, they have reduced growth, reduced feed efficiency, reduced protein efficiency. So at one point, it's maybe keep increasing the litter size is not a good strategy anymore if we have 20-30% of the piglet of the litter that is affected by this condition. Thank you.
Some people are typing in the chat. You can wait for the questions there.
Thank you.
Okay. Is there any other questions? You can type in the chat or just speak with the microphone. I think nobody has a question
fine,
anymore.
thank
Oh,
you.
Carolina. Yeah.
No, I'm fine. Just say thank you. Nice presentation, and hope to see you
Thank
all
you.
next. Yeah, thank you.
So thank you for your presentation. It was very interesting, and good luck with the next step in your PhD.
Thank you, and thanks a lot for inviting me.
Bye.
Bye, have a nice day.
Bye,
Bye.
bye.